Doesn't get the.
Good morning, everybody. It's with great pleasure that I'm introducing Florence Pradillo and Marianne Cambon to give this talk about the blind shrimp, Rimicaris exoculata. Marianne has got her PhD on studying deep sea thermophilic archaea, and she's working on IFREMER since 1997. She has been developing several approaches to study microbes on the deep sea, and more recently, she has been studying the association between microbes and vent animals like the blind shrimp and polychaetes. Florence Pradillon, she has done her PhD in Paris at the team Amex, and then she moved to Germany and to Jamstec in Japan to do postdocs about developing methods to identify larvae from the deep sea, and also to study colonization and connectivity among other chemosynthetic environments. She's in IFREMER since 2010, working on the Laboratoire d'Environnement Profond, and she's been working on Rimicaris life cycle at mid-Atlantic reach. And with no further delay, I give the words to Florence and Marianne. Thank you for joining our REACH seminars.
Thank you very much, Anna, and thank you to the organizers of this series of meetings for inviting us to give this presentation that we will both, Marianne and I, give together, sharing some parts of the presentation. So as Anna said, we will today talk about the emblematic vent species Rimicaris exoculata, which is a model species in our laboratory that we are using to tackle different questions that we have advanced, particularly regarding symbiotic relationships and life cycle of deep sea species. And we should acknowledge that this, the work we will present involved also many of our colleagues that we have mentioned a few of them, those who have participated most in the studies we will present. So I will start with a brief introduction on Rimicaris and its environment, and then Marianne will talk about the symbiosis of this species, and I will get back to give some information about its life cycle. So first, as a short introduction, I will just talk about the context. So life at hydrothermal vents, he's seeing very extreme condition in our view of human, of course, because for some of the organisms living there, these conditions are the normal conditions. So animals at vents face high pressure, as most vents lie on the deep sea bed. Vents are also environments that are unstable, both in space, as they are island habitats that are separated by numerous high space, which are not with venting activity. They are also unstable in time due to the limited time that fluid venting can lengthen in time. They experience strong geochemical gradients, and they are often exposed to fluids that are loaded with toxic compounds. However, at vents, most of the time we see high biomass living communities with dominant species and other species that are highly adapted to this type of environments. So one of these dominant species are the shrimps, Rimicaris, and it belongs to the family Alvinocarididae, which is a family that is endemic to chemosynthetic ecosystems, such as hydrothermal vents, but also cold seeps. And they have a worldwide distribution. I'm showing here a map where you can see all the species that are currently species with names. So you see that there are many species. Of course, this is moving all the time. Some species were found in different environments, in different places, and later realized that they were only one species. This is, to our knowledge, rather up-to-date list of species. So for Rimicaris, exoculata inhabits on the mid-Atlantic ridge. You can see here its geographic range. And it co-occurs with 4 other species, especially in the northern part of the mid-Atlantic ridge, which you can see have very different morphology. Rimicaris is characteristic by its enlarged cephalothorax. So the head is quite large, and this allows the housing of symbiotic bacteria. You can see that other species are very different. Rimicaris fortunata is much smaller and doesn't have an enlarged cephalothorax. Albinocaris markensis is also very different, and they inhabit different parts of the habitat on the vent fields where they coexist. There are also, for Albinocaris markensis, it is also known from other vents on each part of the mid-Atlantic ridge on the side, on continental margin. And it is known as Albinocaris muricola. And this is an example where you have different species, the same species inhabiting different habitats that get different names. And it also highlights that there are some dispersal and connectivity issue that are still moving in this family. And I also wanted to mention the shrimp Rimicaris hibisi, which lives on the mid-Cayman rice, which is very similar to Rimicaris exoculata in terms of ecology. It has also an enlarged head and lives in large swarms like Rimicaris, but it's much more closely related in terms of phylogenetics to Rimicaris chassei. And this also questions the way these shrimps have colonized these vents and how they have adapted to their environment and developed the symbiosis. So Rimicaris is the dominant species on many of the vent sites on the mid-Atlantic ridge. You have here a map with showing the main sites where it was observed, with high abundance at vent fields such as stag and snake pits, but they are also abundant in Logachev and Rainbow, and in the Dayin site on the South Mar. They can reach high densities, covering vent chimneys to the extent where you can't anymore see the substratum below the shrimps. They showed potential high dispersal. Some studies on their genetics have shown that the genetic variation is not varying between vent sites, which are quite far away. And this suggests that these species can migrate from one site to another quite heavily. But studies are still underway, some of the studies. And this is a species that really lives at the edge of vent fluids. They are often seen crossing through hydrothermal plumes, although they don't stay here. And often you can see, such as you see here on the picture, shrimps that have burnt in their carapace. It's quite often, and really shows that they live very close to the fluid exits. So Rimicaris is living on vents with contrasted geochemistry. So here you have the characteristics of the end member fluids, so the undiluted fluids of the vent sites where they live, and they can live on sites which are hosted on the mantle rocks, which gives to the fluid a specific signature. For example, at Rainbow, where the iron concentrations are very high, as well as hydrogen and methane, whereas sulfides are a little less concentrated, and on other sites, such as Stag, which is hosted on basaltic rocks. So contrasted geochemistry. And for the shrimp themselves, this was the M members. I made here a small summary of the information we have, because the measurements really within the environment of the shrimps are not so numerous so far. And most of the time, the aggregates are at temperatures that are between 3 and 30 degrees. Most of the time, it's even below 20 degrees, with a near neutral pH some sulfide, methane and iron can also occur within the aggregates, and with oxygen concentration, which can be a little bit depleted compared to the ambient seawater. So due to this location in its habitat, Rimicaris has been a lot studied for its adaptation to its environment, in particular to temperature adaptation, which is a work that is carried a lot by our colleagues in Paris, in Bruce Chilito's team with Juliette Ravo and Magali Binden. And they've made a lot of studies using high pressure vessels that they developed, and where they saw that exposing, they could do experiments and expose shrimps to different temperature conditions. And this is a summary of all their studies showing that the shrimps mitigate their environment through both their behavior and also through the synthesis of specific molecule. So this is called the heat shock response. These are proteins that can repair proteins in the cells when the animals are exposed to temperatures that cause damages. So you can see on this graph, this is a summary of how the shrimp behave and how they react to different temperature. And you can see that above 20 degrees and until 30 degrees, they mostly use behavior to cope with the environment, and above 30 degrees, which can be something which is lethal for the shrimp when it's exposed for long duration. You start to have a really strong behavioral response, as well as expression of heat shock proteins or cell repair expression. And however, the shrimp can cope with some short-term exposure to high temperature, which will elicit a heat shock response also on the short term. And this also is going with what we see in the environment when we see shrimps that have burns, that really we know that they will go very briefly through the high temperature fluids, but they cannot stay there for a long time, and their normal environment would be more temperature below 20 degrees. So I will now just show you a video, which is a compilation of sequences that have been recorded with the no-till during the cruise at Tag Venfield, which is the cruise because that Marianne will talk a little bit more about later. So you can see very dense swarms of Rimicaris exoculata on the wall of the chimneys, with which are very active, trying to go into the fluid. Here you can see that they are really very close to that. You have different life stages. Here it's maybe not less obvious, but also they share their environment with other species. You have here the red small shrimps, which are the juveniles. You can see here some gastropod Peltospira smaragdina that are sharing the environment also with Rimicaris. Here you see the Rimicaris chassei and Mirocaris shrimps that also occur with Rimicaris exoculata, but more at the periphery of the big and high density swarms that you can see here. So here you have almost only Rimicaris in high densities, and at the edge you have also the other species that occur. So here you see the edge. You see on the center many specimens with burnt, and this is also at the edge many specimens in lighter color, which are recently molted shrimps. And with that, I will leave the floor to Marianne, who will continue with the symbiosis.
Okay, so thank you, Florence. So let's move to the symbiotic part. So Rimicaris exoculata, as Florence said, have catch the attention of scientists when she was discovered, because it was as if it was able to eat chimney particles and the sulfides But the question was first how this shrimp can feed, and looking inside the large and enlarged bronchial chamber, scientists first observed dense filamentous bacteria covering all the interior part of the bronchostegit, which is the carapace here, but also two mouth parts that are beating for the flow of seawater and that are enlarged also and completely covered by bacteria. So the hypothesis was how the shrimps could live in so dense aggregates around the chimney walls. The shrimps still have a stomach and also a digestive system open, so it could feed normally as coastal shrimps. So three main hypotheses were shared when we started to look at the shrimp more densely. That the question was the shrimps was able to graze their symbionts located inside the bronchial chamber, or the shrimp could eat bacteria that are colonizing in dense mats, chimney sulfides, and that's why we could find some minerals inside the digestive system, or would the shrimps have a digestive microbiota dedicated to nutrition, or maybe the three of them. So the question about the shrimp nutrition, because it is very dense population, very active, and these shrimps is, as all crustaceans, molting very intensely every 10 days, so needs a lot of input in a trophic supply. So in the dark, in the deep sea, there is no light, so it's not possible to rely on photosynthetic supply. Instead, we have the role of bacteria that are able to use reduced chemical compounds from the fluids, such as sulfur, which was quite known at the moment, but also hydrogen, methane, or iron, and so on. And the question was how these shrimps could use some reduced compounds, and would these bacteria associated to the head of the shrimps would be implied in the nutrition. First, the shrimps were supposed to have only one single symbiont, here in dark orange. Hope you can see here. So the first symbiont described was here. It was not possible to cultivate it in a laboratory, but it was supposed to be a sulfur oxidizing symbiont. When we first look at the diversity in the bronchial chamber, in fact, we find that it was much more diverse than we thought, sharing not only one so-called epsilon proteobacteria, but several lineages, but also some gamma, delta, alpha proteobacteria, some known to be linked to sulfur cycle, but also to methane or iron. So this opened much more hypotheses about nutrition and adaptation capabilities between contrasted geochemical sites, as Florence presented before, such as rainbow, which is ultramafic, or tag and snake pit, which are basaltic ones. So here, just for illustration, a picture in microscopy, you can see using a chromophore. In green, you have the epsilon proteobacteria, which are now renamed Campylobacteria. We have several morphotypes. And in green, you have the sulfur oxidizing, and some may also use hydrogen. And in red, you have the gamma proteobacteria. So we looked deeper to see whether or not we could have several metabolisms shared in the bronchial cavity. We look at the APS reductase genes. We are implied in sulfur cycle and find several symbionts who share the genes, and some of them were expressed. So we know now that sulfur is probably used. Then we looked for methane because we observed here circled in green, we observed some bacteria with intracellular membranes, which are typical from methanotroph bacteria. And when we look at the gene, we find the PMOA, which is a gene implied in methane captation in the environment for bacteria. And also we could observe them like donuts, as we can see here in green on the picture, which is typical for methanotrophic bacteria. Then about iron. In the deep sea, where the shrimps are living, there is oxygen because the shrimps are doing respiration as us. So oxygen usually is not good for iron oxidation because the iron will precipitate immediately. But in the shrimps, when looking a bit closer to what's happening, here you can see that the seawater flow enters the bronchial cavity in the number 1 here, and then go to the bronchial part for respiration. So there is an uptake of oxygen and a release of carbon dioxide, and that the flow goes out on the third part, where we can see a lot of iron oxide and bacteria embedded in iron oxide. We then try to cultivate some of these potential iron oxidizer using a gradient between substrate releasing iron and on the top of it oxygen. And we managed to enrich but not isolate any proteobacteria implied in iron oxidation, but we can observe them with a dedicated probe. Here in green on the picture, they look like small curved rods associated to the shrimps, and they were identified more recently as belonging to Zeta proteobacteria, which are only known now as free living bacteria occurring in lowy cement, for example, on iron mats. And it is the first time it's described by the symbiont. As we said before, the digestive system is still open, so the shrimps is able to eat something anyway, but the stomodeum, the stomach, is quite reduced compared to the shrimps. So we also looked at the diversity of bacteria inside the digestive system, and we find some campylobacteria which are close to the one from the bronchial chamber, but also for the first time, we found mollicutes and Deferribacteriales, which were not isolated or described in deep sea environments, but their role are still enigmatic because after starvation experiments in the deep sea aquarium, the mollicutes and Deferribacteriales are still present in the digestive system, so they are really attached to it. Using microscopy, we observed here on the slices of the digestive system that it's full of mineral, but we can also here see long filaments occurring inside the digestive tubes. And using SEM, we can see like spaghettis. And using TEM, we can see here that bacteria are just inside between microvillosities of the epithelium of the digestive system. And looking at using fish analysis using probes, we can see that we have active bacteria, long filamentous active bacteria. Here they are in red, and in blue it's a nuclear cell of the shrimps, the nucleus of the shrimps. And so these bacteria, whose role today are still enigmatic, are active, alive, and they are maintained in the digestive system after starvation periods. So they are really associated to the shrimps, but yet it's difficult to know what are their role. The last point to prove that we have a trophic relationship between the shrimps and its bacteria was to look whether or not these bacteria are active in the system, and are they really fixing carbon, and is the carbon transferred to the shrimps, and how. This was a work done by Julie Pansard and colleagues from Paris using the pressure-visual aquarium hippocamp on board. So we incubating bacteria alive with the shrimp alive, supplemented with radiolabeled carbonate, and supplemented with sulfur and iron as electron donor in order to make the symbiosis, the chemosynthetic symbiosis active. And then after a period, we can see that the bacteria are stained by the radioactive material. And if you wait a bit more, you can see that the membrane of the shrimp is also labeled, but not the one from the digestive system, but rather the one directly of the membrane in the bronchial chamber. So this shrimp is a shrimp that is eating by the head, the organic matter that is synthesized by the bacteria is directly transferred across the bronchial chamber integument. Recently, we have done a more complete approach to try to reconstruct the genomes of the symbionts, because these symbionts are still not cultivable in the lab. So we cannot study completely their physiology and their capabilities in terms of adaptation, in terms of colonization. So we have extracted all the DNA of all the bacteria in the bronchial chamber of shrimps from tag and rainbow, which are contrasted environments. And we have reconstructed 49 metagenomes, which are more like genomes from a group of bacteria very close the one from the other. And 16 were more than 90% complete, which is quite good. And we can see that we have there 2 Zeta proteobacteria genomes. And the result we just obtained, the paper is now in press, is that even if many symbionts co-occur in the bronchial chamber, each of them have a special niche, and they are able to cooperate between them in order to produce amino acids, lipids, sugar, vitamins, proteins, that is able to have a complete trophic supply for the shrimp. I have made a small summary on this cartoon. So we have the shrimp exoculata here, and the 4 main bacterial lineages associated to the shrimps. So in red, the Zeta proteobacteria, which is fixing carbon dioxide through the CBB cycle, as the desulfobulbus, which was before called the Delta proteobacteria, and the gamma proteobacteria, so blue and green, and they are all fixing carbon dioxide. They are able also to use nitrate. Several of them are able to use hydrogen. For the first time, the Zeta proteobacteria, we find the CYC2, which is able to use iron 2, and in yellow, the Campylobacteria using the air-TCR cycle, which is another way to fix carbon. Very effective also, which is the main symbiont, also able to use hydrogen. And some points still to be cleared in the next future is about acquisition and exchanges from the shrimp to the bacteria, which is for the moment less known, and the communication between all these populations, both in terms of bacterial communication and host symbiont communication. So as I said before, the bacteria are colonizing the bronchial chamber, such as a biofilm. And we looked at the possible symbiont communication, how they are occurring in niches, and particularly in terms of trophic systems, they are able to cooperate. And in fact, we also find that the chromosomal genes, such as LUX-S or LUX-R, which are implied in biofilm formation and bacteria communication, are retrieved in the genomes of several lineages. And these genes are implied in a communication where when a threshold of cell density is obtained, then the molecules are excreted in the medium and stop the bacterial proliferation. And one hypothesis is that when the bacterial proliferation is stopped, this may be a way to communicate also or interact with the shrimps along the mold cycle. The shrimps is normally also able to communicate with the symbionts to avoid proliferation outside the bronchial chamber. So we have found a small part of gene in the shrimp genome, which is crustine. So the RE for Rimicaris exoculata, crustine, is an antimicrobial peptide. Usually, the antimicrobial peptides are synthesized by the host in order to defend against pathogen. So usually, they are used to kill bacteria. Here in this paper, we have observed that the the antimicrobial peptide is not an agent to kill bacteria, but instead it seems to attract them. And here they are colored in green with the antibody system. And what we can see is that the bacteria are embedded in the erythrocystine, both on eggs on the left picture or at the adult stage on the right one. And so this is probably a way to communicate and attract symbionts and to select them in the environment by the shrimps. Well, still we have many questions to go on with this model of shrimp and this model of communication and adaptation in the deep sea environment. So for these purposes, we have cruises dedicated to the study of deep sea ecosystems and a focus on these shrimps. These cruises are called BICOS for biodiversity, interaction, connectivity, and symbiosis in extreme environments. So we have done the 1 and 2, and now we are going on writing the third one. So the BICOS cruise series occurs on tag and snake pit sites, here pointed in red. We are using the Nautil and the Hervé Pourquoi Pas, which is the biggest oceanographic boat in France. The main tasks are to explore the area between tag and snake pit, to describe the biodiversity of these ecosystems and how they function. We also have a point of the life in the periphery around the vents. Of course, a large part is about life cycle, which is poorly understood now, and the connectivity and dispersion between the different active system. So these cruises are really pluridisciplinary studies with a cartography for map at high resolution to better understand the topography and the system. We also have an imply in geology and geochemistry to better decipher with habitat condition. And then we can better approach the biodiversity of animals and fauna in general and to replace it in habitats and maps. We have a point on microbiology, both in terms of diversity and adaptation. The symbiosis we have talked about. We have a point on adaptation, both pressure adaptation, physiology, temperature adaptation, and of course, life cycle and connectivity. Florence will talk to you now.
Thank you. So now, of course, get back to the shrimp. The life cycle of this species is like for many other species, advances in the deep sea is still enigmatic. You have here drawn what was known before, more or less before the BICOS cruise. We know that adults occur in very high densities advanced, and the juveniles were described as these orange shrimp, which were initially described as a different species, but we know now using genetic markers that they are shrimps of Rimicaris. And this species is forming very high density population, and we know from genetic that it is able to disperse widely. So that would mean that they would produce migrants and larvae in large number, but for a long time, reproduction of the species was not observed. So Rimicaris is a Caridian shrimp, and this is the typical life cycle of a Caridian shrimp. This is the example of the red cherry shrimp that we raise in our aquarium, where you have mating, which is preceding the spawning of eggs by the female, and the females will keep the brood under its abdomen between the small legs that you see here, which are the pleopods. And the female keeps this brood until the embryonic development ends and release small larvae, which have several stages in the water column and then comes back as juvenile and to grow in adult populations. So for Rimicaris, for a long time, brooding female were not observed until in 2007, during the Serpentine cruise on the Logatchev fan site on the Mid-Atlantic Ridge, which occurred in March. where some females were collected brooding eggs, and there was so the first observation of eggs and just hatched larvae for the species. Then question remains on what is the fate of these larvae, how they disperse in the water column. There were some studies where nets were towed around vents, where some juvenile shrimps were collected, and they were identified as albinocaridid shrimps and Rimicaris shrimps, but at that time the genetic identification was not as pushed as we do now, and the species was not the identification was not made at the species level. And then when the larvae settle, you have different the juvenile stage that were described, and the life cycle between benthic and pelagic phase is close, but you see that there are many questions, and one of the big questions for this symbiotic species is how the symbiont acquisition is made and how it is transmitted through the life cycle. Is it the eggs that are inoculated with the symbionts, or is the symbiont acquired later when the shrimp return advance as a juvenile? So I will first talk about reproduction. So before our cruise, most of the studies that were done on reproduction, well, the few studies done on reproduction of Rimicaris, were looking at the ovaries, which are here the red parts, which are the eggs before, when they develop inside the body of the mother, before they are spawned and held between the pleopods. So the studies were doing some microscopic analysis, doing some section through the tissues and looking at histological cuts. You can see here on this histological section the eggs that were measured, and with that, the developmental stage of the egg was analyzed. So you are here studies by Copley where they looked at the mean diameter of eggs in different shrimps, and this is representing the mean diameter. And what you can see is that you have each box plot is one individual. So in a population, you have females with eggs at all developmental stages, and these were observed at two times in the year, mostly in autumn. And with this type of pattern, usually we conclude to a continuous reproduction because you have females at all stage of gamete development. And this also continuous reproduction is often the main hypothesis for deep sea species, where seasonality may lack some cues that would impulse this seasonality. And since ovigerous females, so the next stage when the female has spawned, were almost never observed, the main hypothesis at that time, since we believed that reproduction was continuous, was that if we couldn't find them, it was because they would migrate in other areas, not in the dense population area, but at the periphery where conditions were milder and which would allow the brooding of the eggs without exposing them to harsh conditions. And this was also, such pattern was observed in some vent crabs. So that was an hypothesis that was quite strong. But in March 2007, some ovigerous shrimps were collected at Logatchev, and they were collected in dense assemblages. So it was the first time that the hypothesis of brooding at the periphery of vents was challenged. And these are images of the shrimps that we collected during the first Bicos cruise in January 2014, where you can see that we had many females with ripe gonads, as you can see here with the pink red color on their back. But we also observed a good number of ovigerous females that were bearing eggs under the abdomen, that you can see here. So this observation, I will not go too much into details, was repeated in two other cruises that were conducted in the winter time in 2017 and 2018. And this scheme is showing a historical record of the cruises and of the collections that were made. And you can see that most of the collection were made during the summer and fall periods. And in this collection, which were made in the dense aggregate of shrimps, there were very rare occurrence of ovigerous females, whereas for the recent cruise that we tried to have in the winter period, we could find a good number of brooding female, which represent about until to up a third of the sexually mature females. So this shows that there is a possibility of there is a temporal variation in reproductive activity in this shrimp, and we don't see a pattern of continuous reproduction as it was believed before. And this is pointing at a possible seasonal pattern, although currently we are still studying this reproductive rhythm to try to find which could be the possible cues for to maintain such seasonal pattern. Another thing about this brooding female, that we saw is that we found females with developing embryos in their brood at all developmental stages of the embryonic development. So you can see here eggs that were just spawned, they are not divided. And then they start development and start cell division, and then tissues will start to differentiate. So you can see here the structure of the shrimps that start to develop while keeping a good amount of lipid reserves, and until a young larvae that is here about to hatch, where you can see the eye here with the eye spot here, the lipid reserves on the dorsal side, and the appendages that are curled around the larvae here. And this is a larvae that just hatched. And all these stages were found bared by females that were in the dense aggregate exposed to the warmer part of their habitat. So these warm temperatures are questioning whether this condition would accelerate development, which could, on the other hand, be more slow if the females were brooding at the periphery. But we saw that for Rimicaris, that was never the case. We never found a female brooding eggs out of the dense population of Rimicaris. It also raised the question of the tolerance to food of this early life stage that could be more vulnerable with less protecting structures, and also questions the possible microbial development on the eggs, which usually in shrimps in aquaculture is sometimes problematic. When bacteria develop on eggs, it can lead to failure in the development. So we've been looking at the bacterial development on the eggs. So you have here on the top row different brood at different stages, which are the stage I just showed, with the early ones which are just dividing, and then mid one which start to develop some features, and the late eggs where the larvae is almost completely formed. You cannot see this in this picture, but you can see that some color is developing, and this is mineral precipitation that progressively embeds the eggs, like it does in the branchial chamber of the shrimp. And here in scanning electron microscopy, you have images of the envelope, and here are the links of the mucus that is linking all the eggs together in the brood. So this part. In early stage, it's almost devoid of any precipitation of bacteria. Here in the mid stage, you start to see some mineral precipitation, but also bacterial development, rod-shaped bacteria, as well as filamentous bacteria. Here is, you see the envelope has several layers, and sometimes it's broken. So we can see that the layer more inside are less colonized. And this is a late stage of development where the mineral precipitation as well as the bacterial development is really high. So there is a trend of increasing mineral precipitation and bacterial colonization along the development, which goes with a prolonged exposure to the fluids, to the external environment. And then we've been looking at the composition of the bacteria that were covering the eggs, with the question of trying to understand whether these bacteria would be specific ones or it would be just fooling from the environment, since we had a trend of increasing bacterial deposition with time that could also be just fooling. So we've been looking at both eggs here and the pleopods, which are the legs here that are holding the brood, so that are exposed to exactly the same environmental condition. And we were thinking that if we would find exactly the same community, then probably most of the bacteria on the eggs would be just fooling. So here you have the result of the analysis of the diversity of this bacteria. With in pink, you have the eggs. In green, you have the pleopods. And we've been looking at mothers or broods on mothers from tag and snake pit, which are denoted by the orange and snake pit by the blue. And round triangle and square symbols are for the developmental stage of the broods. So when you look at here, you have the diversity of the symbionts that were found on the pleopods and the eggs. And the communities are dominated by, in orange and yellow, by delta and gamma proteobacteria, but you also have a large amount of epsilon bacteriota, which are of different types. And what you can see is that these epsilon bacteriota are dominant on pleopods, but are also found on eggs. And what you can see is that the dominant light blue one is belonging to the Sulforovacea, which are sulfur oxidizing filamentous bacteria that are found both in abundance in pleopod and in eggs, and which can also be found in the environment. And here in the medium blue, what you can see is that you have a different type of epsilon proteobacteria, which are the Campylobacteracea, which are a lineage that is found among the symbiotic community in the adult, in the cephalothoracic cavity. So you have on the eggs, more specifically, this lineage, which is implied in the symbiosis in the adult. And another one, which is worth noting, is the small molecules, which are here in light green, that are also more found on the eggs and which are found in the digestive tract of the adults. So with this analysis of the diversity of the microbial community on the eggs, we could have hints at the fact that this community might show some specific patterns on the eggs, which include some symbiotic lineages and which may provide hints at the fact that maybe symbiosis, something on the symbiosis, on the acquisition of symbionts could occur at these early stages. And this was, so this is an image of a late stage egg, where you can see here labeled with fluorescence in situ hybridization probes the bacteria, which means that they are active community, it's not dead ones. And I come back to what Marianne just showed on the synthesis of the antimicrobial peptide crustine, which we show us is synthesized by the egg. So in early stage eggs here that you have here on the slide, it's difficult to see, but you can see some fluorescent labeling of this antimicrobial peptide, which is here on the envelope of the egg at early stage. And here is a late stage eggs where you also have this anti-microbial peptide that is in vesicle within, below the envelope of the eggs, but which is also found embedding the bacteria at the exterior, on the external side of the envelopes that you have here and here in large view, and which is pointing at perhaps a mechanism of recognition at this stage of the bacterial symbionts. So now we've seen that the eggs were brooded in the vent fluid, and this may have implication for both development and also symbiont transmission. Now the question is where are the larvae traveling? What's their fate? So to better understand that, we need to collect not larvae that are just hatched from the female, because this doesn't give us many information about the larval life, but larvae that would be collected after hatching in the water column. And for that, we've used an in situ larval pump that we developed in the lab that is pumping water in the surrounding environment and is trapping the larvae and any particle from the water in sampling bowls. So with that, we've collected such planktonic particles around vent sites near tag and snake pit, and we've collected a good number of larvae, among which we had some very early, some stage of larvae of Rimicaris exoculata that we have identified using genetic markers. But what you can see is that these larvae resemble very much closely to those that just hatched. So they are very early zoe larvae, not later stage that would give us information on the pelagic life of the larvae. All that said, we still could get some information with this early larval stage. This is a picture of the zoe larvae in scanning electron microscopy, and what we could see when we are looking at the moth part is that these moth parts are relatively not developed. Here is a comparison with another shrimp, a Caridian shrimp species, Macrobrachium, showing the same part. So what you can see is that they are rather simple for the mandible, and they have some CT. But in this shrimp, this type of development was related with planktonic larval mode, which is called lecitotrophic, which is the term describing larvae that are not feeding, but are developing from the reserve they have and they carry it with her. And you can compare that with another type of larvae that has a mouth part that are much more developed and armed with spines and CT, which allow them to truly capture particles to feed themselves. And this is called planktotrophic development. So for the larvae of Rimicaris was before believed to be a planktotrophic larvae, but what we see is that the early larvae, when it hatched, is not able to feed at the beginning and is more developing on a lecitotrophic mode from the reserves it has in the cephalothorax part. So this is just a reconstruction of the different larval modes that are known in Caridian species, for which the early stage of larvae are known, where usually when the larvae hatch, there are two main types. Either they have moth parts that are well developed that allow them to feed, but the legs, peropod and peropods, are not developed. So it's a quite early stage of development. It needs much more development, and these larvae are usually planktotrophic. They develop for a long time in the water column and they feed themselves. And the other type is the larvae that hatch with large reserve, undeveloped mouth part, but that are well developed in the rest of the body and usually have a development that is relatively short, larval period that is relatively short. And these are the two main types. And here is just, it's a maybe not so common type, but where you can have some small differences in the different features. And what we found with the larvae of Rimicaris, but we also compared with other albinocaridid species, is that we have both undeveloped month part, which is going with less heterotrophic larval mode, but we have relatively also the pleopods and periopods were relatively not well developed, which would fit more an extended larval period. So it's a combination of trait that is not usual for deep sea caridian shrimps. And this is just a summary showing you that these are the two main types I just mentioned, with some example among vent animals and crustaceans, where you have either larvae which hatch at an advanced stage and which have lots of reserve, which do not feed, and this goes with a short larval life and with not a strong dispersal and rather local retention near the birth sites, contrasting with larvae that hatch with the possibility to feed with low reserves and which would have a much longer larval period and would disperse much more. And for Rimicaris, we have a combination of lecithotrophy, which would occur early in the first zoe stage, but still we have signs that it needs a long time to develop. And this is also going with a Plantar trophy that would perhaps develop after the first zoe stage, but at the moment we only have this, and which would be compatible with high dispersal. But at the moment, that's what we can say from this larvae. We still lack a lot of the stages that are dispersing in the water column. But what we have is the juveniles, and that's what we also looked at to get information about this, both the life cycle part that is before settlement and what's happening after. So during the Bicos cruise, we've observed something that was already mentioned in papers, but we observed nurseries of juveniles, both on the column, on the chimneys, very close to the assemblages of adults. But also we observed some nurseries which were on the flat bottom near diffusions and not near to the adults, which are here. And within these nurseries, we found different type of juveniles, different sizes that we've tried to identify using morphological characters that were published. But this revealed difficult because some of the characters occurred on different of the stages, and morphologically it was very difficult to distinguish the species. So what we've done is using genetic to identify these juveniles. And what we found is that the juveniles, I'm going back to that were found on the flat substratum, were juveniles belonging to Rimicaris chassae, the other species living in these environments, whereas those living on the chimney walls near adults were juveniles of Rimicaris exoculata. And this juvenile that was earlier described as the first juvenile stage of Rimicaris exoculata was in fact Rimicaris chassae. So this led us to revise a little bit the taxonomy and the species definition in early stages of the two species, with 3 juvenile stages for Rimicaris exoculata and 2 for Rimicaris chassae, and not 4 for exoculata and 1 for chassae as it was published before. So with this redefinition of the early settling stage of Rimicaris, we have looked at some hint of the pelagic life, looking at isotopes as it was already done before, but using this time really well identified each stage of each of the two species. And this is the result of this isotopic analysis where we've looked at both carbon, nitrogen, and sulfur isotopes. And here we are only showing the carbon and sulfur. So the carbon is providing information about the source of nutrition with nutrition that is based on chemoautotrophic symbiosis, which rely on the CBB cycle with signatures that are well below minus per mil in carbon, whereas chemosynthetic, no, okay, this is for autotrophy and CBB, and the Artesia cycle, which is made by epsilon proteobacteria, has a signature which is very different. So the signature, the source of the carbon is given by the carbon isotopic signature, whereas the sulfur was used as a marker of chemosynthetic or photosynthetic origin of the nutrition, because the in photosynthetic pathways, using sulfate, the signature gives value that are much higher than those using sulfide oxidation in chemosynthetic environment. And what we saw is that for the two species, we have a gradual trend of nutrition, which is based on what is thought to be a photosynthetic origin in early juveniles for both species, although there is a difference between the two species, shifting progressively in adult stage towards a nutrition which is more and more based on chemosynthesis, which was already known, but here we see that there is a difference between the two species, which may also reflect a small difference in nutrition during the larval life. And what we should add is that at the moment, this photosynthetic origin of the nutrition of larvae is based on what we know from the signature that are given by a photosynthetic pathway, but we lack studies looking at microbial productivity in the water column in the deep sea that could produce, because they also use sulfate, a signature that would also be compatible with what we see with juveniles, and which would perhaps provide us different hints at the possible places where larvae would disperse. They are not necessarily eating some photosynthetic material that is sinking from the surface, but perhaps they are also grazing on bacteria that develop in the water column and that would provide a similar signature, but this really is something that needs to be looked at in the future to have better idea of what is the feeding strategy of these juveniles. So just to conclude, we've presented many aspects of the life cycle and symbiotic relationship of this shrimp. But as you see, that there are many, many questions that remains open and that are very important to understand what is the life cycle of this shrimp and what would be the sensitive stages in a perspective where this habitat would be impacted, then we would need to know when the colonizing juvenile arrive, when the female reproduce or the population reproduce, and when the larvae are at risk of being exposed to some challenge in the water column when they are dispersing. So we have many questions open on the reproductive rhythm and the potential cues that are possibly triggering seasonal reproduction. We still have questions on how the brooding duration would impact symbiont interaction. Of course, since we still lack the larval stage, we have a question about the swimming ability interaction with water masses, which would have a strong impact on the dispersal and connectivity of the species. We also need to understand better how the larvae orientate and find the sites where they settle. and how the symbionts may be acquired. We've seen that possibly some of them are on the eggs, but at the same time, we've seen that the larvae are unable to ingest something when they hatch. So which question really the symbiont interaction in egg, but possibly the acquisition would be at the arrival of the juvenile. And then the potential role of these symbiont acquisition in adult metamorphosis. And this we are looking at, we would like to look at this question by comparing the Rimicaris exoculata with its large head and strong metamorphosis with what's happening with Rimicaris chassei, which has not this metamorphosis, but which is very closely related to the species Rimicaris hibissae, which also has the metamorphosis of the big head, although it's very much more closely related to Rimicaris chassei, but it occurs at a site where it's the only Rimicaris species. So it's pointing at potential interactions between the species for trophic resource that would drive or not the development of the symbiosis. And with that, I'm finished, and I think we are happy to take any questions.
Thank you.